Good morning, all the students. First of all, I would like to congratulate all the students for coming into the second year. So I will be taking this course of APB 5121, 5122, sorry. That's the name is Principles of Plant Breeding. So today from now onwards, we will be taking your classes through Zoom app. So kindly, all the students have to download the Zoom app and the Google Forms. So we will be giving some questions and you have to answer those questions to the Google form. So today my topic is about the history of plant breeding, the objectives of plant breeding, and the major achievements that have been achieved in plant breeding. So today, this we will be covering. Okay. So through PowerPoint presentations, uh, I will be showing you the slides, and for the next 20 minutes, I will be covering this topic. After that, I will take all the questions. If you have any doubts, then I will be clearing all your doubts. Okay. So we will move to the next slide. First of all, introduction to plant breeding. What is plant breeding? Actually, the plant breeding, it is an art and science. Means it is art also and it is a science of improving the heredity of the plants. We are in plant breeding. We are trying to improve the heredity of the plants. That means hereditary characters of the plants. That is ultimately beneficial for the humankind. So the definition we can see is it is the art, plant breeding is the art and science of improving the heredity of the plants for the benefit of the humankind. This was a definition that was given by J.M. Pohilman in 1959. So we can say that it is an art as well as a science which tells us the means to change the genetic architecture of the plants. Here, the main objective of the plant breeding is to bring about crop improvement or to improvement in the yield of the plant. So we can go for either through agronomic characters, that is, either we can improve, either we can bring about the improvement in the plants through the agronomic characters, that is by bringing improvement in the agronomic characters, or that is final aim is to increase the yield. So, and the second objective we can do by genetic manipulations. Nowadays, plant breeding, we are basically moving towards the biotechnological tools where by using various biotechnological tools like molecular markers, marker-assisted selections, we are changing the genetic architecture of the plant so as to attain the particular objective of the plant. Okay, it has been practiced for thousands of years. So plant breeding is not a new science; it is an old science. We have been practicing this plant breeding from the age-old civilizations when the emergence of the plant breeding when the plant mankind takes place. So it has near the beginning of the human civilization we have been using this plant breeding. So it's not a new science. However, we are day to day we are bringing about some new biotechnological tools so as to change the genetic architecture of the plant. So the ultimate objective is to bring about an improvement in the heredity of the plant. Okay. So this was the definition of the plant breeding, which was given by J.M. Pohilman in 1959. So you can learn different definitions of plant breeding from other books also. Now coming to the next slide where we will be seeing that what are the major important activities of plant breeding. Okay, what are the major activities of plant breeding is first is creation of variability through hybridization, tissue culture, and mutagenesis. So, either through hybridization, you all know hybridization. What is hybridization? When we are crossing two lines, when we are making two crosses between two lines like A line and B line, then the F1 is generated, and F1 is when F1 is taken to the F2 generation, then it is called as a segregating generation. So whenever we do crossing between A and B line, that is called as an hybridization. So there's a particular technique. Okay, this is a particular very age-old technique of plant breeding. Then second one is tissue culture. The various tissue culture techniques also we bring about creation of variability. We do we study creation of variability through mutagenesis also. So mutagenesis means by using various chemical radiation, physical radiation. So this we have studied in the last year course also. Then the second one is selection of elite types from diverse populations. Okay, from the different diverse populations from various land races, like local land races, we go for selection of the elite types. Third one is evaluation of the selected genotypes in the multi-location. So trials. So first of all, third point is evaluation of selected genotypes. Genotypes are generally selected based on their characteristics, and then they are taken to the multi-location trials. And after these multi-location trials, we identify certain superior genotypes based on their testing. Okay, so there are certain seed testing labs and the seed testing uh, testing criteria. So the main aim is to identify the superior genotype. Okay, then we go after the identification of the superior genotype, we go for the multi-location trials. Okay, for the different multi-location trials we go, and then final step is the distribution of the new variety to the farmers for the commercial cultivation. But finally we have selected a particular superior genotype, then we go for multi-location trials, and then we give it to the farmers for commercial cultivation. So this is the main important activity which has been taking place in the plant breeding. Okay. So the broader objectives in plant breeding, we can divide them into three types. That is crop improvement. We try to bring about the improvement in the crop through various agronomic practices or by genetic manipulation. Okay. And the second point is improved agronomic characters. The plant breeders also objectives is to bring about agronomic improvement in the agronomic characters, like we want to increase the grain yield or the biomass yield. Okay, number of tillers, number of productive tillers, number of grains, 100 seed weight. So these are the various agronomic characters which we try to bring about in improvement in them. And the third point is, this is a very important point nowadays, looking into the present scenario, mainly resistance to various biotic and abiotic stresses. Resistance to various abiotic and biotic stresses, mainly, what are the biotic stresses? Biotic stresses are mainly due to the insect pests, diseases, like these are the factors which affect the grain yield, and the abiotic factors like environmental factors. Okay, abiotic factors include the environmental factors like salt, salinity, like various epidemics and pandemics. So these are all the abiotic stresses. So as a plant breeder, we should also try to develop those breeding lines or those superior genotypes which are resistant. Those varieties we should try to breed or to develop those varieties which are resistant to the various biotic and abiotic stresses. Okay, because nowadays this is in high demand. Now we'll see point by point. Now we'll see the various point by point these increased yields and the various points which I have listed above. So we'll see it in detail. Increased yield. Majority of our breeding programs they aims at increased yield. This is achieved by developing more efficient genotypes. Okay, what do you understand by this? Most of the breeding programs which we do, they are aimed at increased yield. That is to develop more efficient genotypes. More efficient genotypes means they are more suitable to the environmental conditions, suitable to the environmental conditions. For example, here the classical examples here we see is DGU gene in case of rice and Norin 10 gene in case of wheat. So this is a very classical example of rice and wheat, which you should remember. So there was a DGU gene that has been introduced from International Rice Research Institute, that is IRRI Philippines, and Norin 10 was a gene that was introduced in wheat. So identification and the utilization of male sterility. So these male sterility and self-incompatibility, this we will be studying about in detail in later on in this subject. Now coming to second point is improvement of quality. So what is the second objective is to bring about the improvement in the quality. For example, if in case of rice, if we see what is the type of improvement which we want. Okay, so that is mainly milling quality, cooking quality. In rice, we always, as a consumer, we always look to the aroma, that is also called as the aroma or the khushbu in case of rice, and that is bought through on. And then grain color, we also see the grain color of rice. Second point is wheat. What are the improving qualities which we look in case of wheat is milling quality, baking quality, and the gluten content. Okay, this stickiness in case of rice, it is mainly due to the gluten content. Okay, then pulses. In pulses, the main aim in pulses is to bring about enhancement in or increase in the protein content, because pulses are the major sources of protein, and improving the sulfur containing amino acids. In case of oilseeds, we always aim at reducing the PUFA content, that is the polyunsaturated fatty acid content. The third point is elimination of the toxic substance. Mainly in case of legumes and linseeds, we always go for reducing. We also aim at breeding those superior genotypes which are in reduced toxic substances. For example, in jowar plants, we always try to reduce the HCN content. We try to reduce the HCN content. Okay. Then in case of Lathyrus sativus, we always try to reduce the beta, that is BOAA content. BOAA, the full form is beta and oxalamine alanine. This is a lathyrosine content, which as a breeder we always try to develop those breeding varieties which are which are having reduced amount of BOAA content. Okay. Then in brassicas or in mustards, we try to develop those varieties which are reduced erucic acid, and in case of cucurbits, we try to develop those breeding lines which have reduced amount of cucurbitacin, that is a toxic substance. Okay, the fourth point is resistance against biotic and abiotic stresses. That we have already told that I have already told that we should always try to develop those breeding, those superior genotypes that have resistance against the various biotic and abiotic stresses. Biotic stresses include the pest, diseases, resistant varieties, that is once we develop those breeding varieties which have pests and diseases resistant, so we try to increase through this way we are also reducing the cost of cultivation and environmental pollution, because if there is pests and diseases, the farmers always spray pesticides and fungicides. So in a way, it is also spreading the environmental pollution. It is also contributing towards the environmental pollution, and thus by using those resistant varieties against biotic stresses like pests and diseases, we are also reducing the environmental pollution and saving the beneficial insects. Okay. And the third point is abiotic stresses. Abiotic stresses mainly include soil factors, salinity factors, frost. So these various environmental factors. So we should also try to develop those breeding varieties which are resistant to various abiotic stresses. Fifth point is change in maturity duration. That is the maturity duration. As a breeder, we should also try to develop the early maturing varieties so that the farmers can have the yield at an early stage, then medium duration varieties and late duration varieties. So the different maturity duration breeding varieties we should also try to develop. The sixth point is improved agronomic practices like improved agronomic characters like production of more tillers. We should those varieties which have more number of tillers. If there are more number of tillers, then there will be more number of panicles, more number of seeds. Okay, so more number of grains. So the grain yield will be more. That is our ultimate objective. Seventh is reducing the plant height to prevent lodging. Rice, mainly in case of rice, those varieties of rice which are tall. So the major problem in this is lodging. Whenever there is, because rice is always there has to be a submerged condition in case of rice. Means the water should be there in the at the up to the tiller level in case of rice field. So the major problem that is in rice is lodging. So we should try to develop those varieties with a lodging resistance. Only the short or the tall varieties are more susceptible to lodging, whereas the those rice varieties which are medium height, they are tolerant. They are little bit tolerant to lodging. And photo insensitivity, mainly in case of red gram and sorghum, those varieties we should try to develop which are not affected by the changes in temperature and light. A non-shattering nature. In case of green peas and brassica, mainly in peas, the major problem is shattering nature. At the time of maturity, these seed grains they shatter. Okay, so there is a major yield loss takes place. In case of synchronized maturity means in case of pulses there should be a synchronized maturity means all the plants mature at the same time. Okay, that will be helpful for the farmer for the harvesting purpose. Then determinate growth habit. There should be a determinate growth habit in case of pulses. That is, they should have a determinate growth habit. It should not be indeterminate growth habit. It should grow straight. Then seventh point is elimination of the and introduction of dormancy. In case of roundness, there should be, depending upon the purpose of the farmers, if they want to store it, then there should be dormancy period in case of, and if they do not want to save it, then we have to go for those varieties. They should not have any dormancy period. Now, what is the scope of plant breeding? Since the cultivable nowadays, if we look at to the scope of plant breeding, what is the scope of plant breeding in the future? Looking into the present scenario, there is considerably shrinking of the cultivable land. Nowadays, we are going for heavy construction. So the cultivable land is day by day it is reducing. So there is no scope for increasing the area under cultivation. So that is the major solution, or the only solution to meet the food requirement is by increasing the crop yield through genetic improvement of the crop plants. So we can only option that is left with us is to bring about certain other ways where there should be always a substitute. There should always be another way for increasing the crop yield. That is either we can achieve through bringing about improvement of agronomic practices or by genetic improvement of the crop plants. Okay, the first one is there are two ways which include the yield improvement is possible. First is enhancing the productivity of the crop. How we can enhance the productivity of the crops? By following various proper management of soil and crops involving suitable agronomic practices and harvesting physical resources. Means we should follow suitable agronomic practices. Thereby we can increase the yield by using high potential crop varieties created by our second point secondly first is through by improving the agronomic practices, and second is by using high potential crop varieties which are appropriate for the genetic manipulation. Genetic manipulation of the crop plants here, how we can achieve through various biotechnological tools, through tissue culture, through mutation breeding, through genetic engineering, through recombinant DNA technology, through plant breeding, through biotechnology. So these are the various ways where we can bring about the genetic improvement in the crop. Right. Second one is stabilizing the productivity achieved. Once the productivity is achieved, but how we can stabilize it? This is also a important point. So we have to stabilize the productivity. This is done by using data especially for wide adaptation of the specific crop genes to offset the ill effects of unfavorable environmental conditions prevailing in the areas. So once the productivity is achieved, we should go for stabilizing. Means if 1 year the productivity is 10 quintals, then the next year also the productivity should be 10 quintals. It should not reduce by it. However, it can increase, but it should not go below that. So that is called as stabilizing the productivity achieved. Now we will see the plant breeding. What is the past of the plant breeding? What is the future? And what is the present scope of plant breeding? Major achievements have been achieved in the past in plant breeding. So Indian agriculture remained stagnant, particularly during 60s. After this Mendel's work, so after for several years it remained stagnant. After that, again in 1900, three scientists again rediscovered the Mendel's work, and thereafter the science of genetics and plant breeding emerged. So breakthrough achievements have been achieved in case of rice, wheat, pearl millet, jowar, and maize. Mainly the cross between the Indica and Japonica cross derivative ADT27. It is the first high yielding rice of Tamil Nadu. So this was the highest yielding rice of Tamil Nadu. This we are studying about. This we are studying about the future, about the past of plant breeding. So the identification now, this is also an important point, that identification of the BGU gene. This was a gene which was introduced in rice variety through IRRI Philippines, and this led to the release of Wonder Rice, that is IR8, that was a cross between Peta and DGWG, changed the scenario. It altogether changed the scenario from poverty to problem of plenty. So this was a breakthrough success achieved in the past of plant breeding. Likewise, identification of Dorfman gene, that is Norin10. Norin10 was a Dorfman gene that was introduced by Borlaug, and breeding of Mexican Dwarf wheat varieties led to the release of, now this gene was introduced into the rice, into the wheat, and it led to the release of the variety that is Kalyan Sona in India. So these varieties, these genes you have to remember. In pearl millet, breeding of male sterile line, that is TIF 23A. This was a male sterile line at Tifton, Georgia by Burton and his co-workers. Later on, its introduction to India led to the release of hybrids, that is this in case of Bajra, it released the hybrid of HB1 to HB4. This was a series that was made possible by the introduction of this variety, that is this male sterile line, that is TIF 23A. In Jowar, the breeding of male sterile line combined Kafir 60A. This was also a male sterile line, and its introduction in India led to the release of first hybrid sorghum, that is CSH1, that was a cross between CK60A and IF84 during 1970s. So this was also a major breakthrough in case of Jowar. At present, we are mainly in search of identifying those sources of phytoplasm in almost all the crops to breed the hybrids with a new source of phytoplasm to prevent the possibility of appearance of the new pests and diseases. So we look into the new sources of phytoplasm from the various sources. Either it can be exotic sources or it can be local land races. So we mainly look into the new cytoplasm sources so that we can introduce into our local varieties, and we can go for developing those varieties which are resistant to biotic and abiotic stresses. So what is the future of plant breeding? future of plant breeding is a challenging task. It is a challenging task because nowadays we are deploying various innovative breeding techniques with a new tool, the biotechnological tool, to assist the conventional breeding techniques. So the conventional breeding techniques have been assisted by various tools. I will say these are the tools, the biotechnological tools. So all together, it is a challenging task uh, in the future of plant breeding. Now we'll look into the undesirable effects of plant breeding. What are the undesirable effects of plant breeding? Since in a plant breeding we are going for hybridization, then we are going for selection, mutation, and the creation of genetic variation. So by creating the genetic variation, we are somehow leading to genetic erosion also. So this is also a problem. Disappearance of land races. Since we are introducing the exotic varieties from the foreign countries like IRRI, Philippines, we are introducing we are not using the local land races. Okay, so the local land races are basically disappearing. So we can say due to the introduction of the high yielding varieties, we are always looking into the high yielding varieties. So in a way, it is leading to genetic erosion of the local land races. So we should also look towards the local land races because sometimes there are certain characters which are present in the local land races which we are looking outside. Okay, so we should also exploit the local land races also. Introduction example is here the introduction of genetic erosion is introduction of IR20 rice led to the disappearance of the local land races of Samba rice. Samba rice is a local land race. So by introduction of IR20, it led to the disappearance of Samba rice. Narrow genetic base. Genetic vulnerability to pest and diseases. In case of Bajra, TIF23 was a male sterile line, but it was introduced in case of Bajra, but later on it was found that it is susceptible to downy mildew. Okay. In case of maize, T cytoplasm which was introduced, it was susceptible to Helminthosporium. So these are also there is a narrow genetic base when minor disease and pests become major due to intensive resistance breeding like rice tumoro virus. So there are minor disease. It is so there are certain minor diseases and pests also. Attainment of yield plateau. This is also a problem where a stage is achieved when there is no further increase in the yield. So there is a attainment of yield plateau, means there is a stagnancy in yield plateau. So in case of Wilmeron, this you have to remember that Wilmeron proposed the individual plant selection, means a single progeny testing, a single progeny from a single plant, that is called as single plant selection. Okay, so he proposed the individual plant selection based on progeny testing. Progeny means whenever two lines are crossed, that one is called as progeny. So this was known as Wilmeron's principle of progeny testing. He proposed this theory testing in sugar content in sugar beets in case of Beta vulgaris. But this method was found ineffective in beet. This clearly demonstrated that there is difference between the effect of selection in cross and self-pollinated crops. So there are different breeding approaches that has to be followed in case of self and cross-pollinated crops. Now we'll look into the history of plant breeding. It stated that if we look into the history of plant breeding is an age-old from the age-old techniques which we are using from the very first from the starting of human civilization. Okay, it's not a very recent science. So it started when man first chose certain plants for cultivation. So there is no recorded history when the plant breeding started. As early as 700 BC, Babylonians and Assyrians artificially pollinated the date palm. In case of 1717, this was the year when Thomas Fairchild produced the first artificial hybrid. He proposed the first artificial hybrid. If you have to remember, in 1717 year, Thomas Fairchild, he produced the first artificial hybrid. Joseph Colruyter, a German, made extensive crosses in tobacco and solanum. He made the crosses between tobacco and solanum between 1760s to 1866 and studied the progenies in detail. So he made a cross between tobacco and solanum, Joseph Colruyter, and then he studied these progenies in detail. Thomas Andrew Knight was the first man to produce several new fruit varieties using artificial hybridization. So Thomas Andrew Knight, T.A. Knight, he in the year 1759, he to 1835, he studied artificial hybridization to produce a new fruit variety. Lee Corsair, a farmer, published his results on selection in wheat in the year 1843. He concluded that progenies from single plants were more uniform. So Lee Corsair, he was a farmer. He studied the results on selection of wheat in the year 1843, and then he studied the progeny from a single plant, that is the single plant selection. They were more uniform. Patrick Sharif, a Scotsman, practiced individual plant selection in case of wheat and oats and developed some valuable varieties. Nelson and his associates in Swedish Seed Association refined the single plant selection. He again studied refined the single plant selections. Then in 1903, Johansen proposed the famous pure line theory. These history points you have to remember that in the year 1903, Johansen proposed the famous pure line theory, which said that a pure line is a progeny of a single self-fertilized homozygous plant. Whenever there is a single homozygous self-fertilized plant, that is called as a pure line. He developed that this theory is based on his studies in Physalis vulgaris. He studied this in Physalis vulgaris. G.H. Schell, he worked in maize, is the forerunner for the present-day hybrid maize program, and he studied about the effect of inbreeding. In 1960, Norman Borlaug, the Norman laureate, developed Mexican semi-dwarf wheat varieties which paved the way for the Green Revolution. So Norman Borlaug led the Green Revolution. This everybody is known. Norman Borlaug for Green Revolution in case of wheat by introducing the Dorfing gene. Dorfing gene is Norin 10. Okay, so this you have to remember that for Green Revolution, Norman Borlaug in the year 1960 in case of why he was given the Nobel Prize, because he introduced the Dorfing gene in case of wheat, that is the Norin 10. And in India, Dr. N.S. Swaminathan and a number of high-yielding wheat varieties like Kalyan Sona, Sharbati Sonara were developed by Dr. N.S. Swaminathan. In rice identification of the Dorfing gene, that is DGU gene, from a tall rice variety by a Taiwan farmer revolutionized the rice breeding. Using this DGWG at IRRI during 1965, the Wonder Rice IR8 was released. So, in case of rice, DGU gene, that is the Dorfing gene, was introduced from IRRI Philippines, and it led to the Wonder Rice, that is called as IR8, was released in India in the year 1965. And last is Nobilization of Sugarcane by C.A. Barber and T.S. Venkatraman is another monumental work in case of plantation. So T.S. Venkatraman and C.A. Barber in case of nobilization in sugarcane. So these points are very important regarding the future and the past of plant breeding. So these you all have to remember these points. By this, I will be concluding my today's lecture. You have to go through all these points, and I would now I will stop my presentation here. Okay, thank you.